Hello everyone. Today in the topic of alcohol production, we are going to study in the subject of industrial biotechnology. So our objective is to study the introduction of alcohol production. The outcome will be that you will be able to understand what are the intrinsic details of industrial manufacturing of alcohol. So industrial alcohol and ethanol. Ethanol, a type of industrial alcohol, also called ethyl alcohol, pure alcohol, grain alcohol, or drinking alcohol, is a volatile, flammable, colorless liquid derived directly from molasses. Okay. Ethanol fuel is the most common biofuel that is used worldwide, particularly in Brazil. Alcohol fuels are most are produced by fermentation of sugar derived from wheat, corn, sugar beets, sugarcane, molasses, and any sugar or starch that alcoholic beverages can be made uh, from, like potato and fruit paste, etc. The ethanol production method used are enzyme digestion to release sugars from stored starches, fermentation of the sugar, distillation and drying. The distillation process requires significant energy input for heat. So ethanol can be used in petrol engines as a replacement for gasoline. It can be mixed with gasoline to any percentage. Most existing car petrol engines can run on blends of up to 15% bioethanol with petroleum or gasoline. Ethanol is also used to fuel bioethanol fireplaces as they do not require a chimney and are fuel-less. Bioethanol fires are extremely useful for new built homes and apartments without a fluid. One of the most remunerative application of molasses is the manufacture of ethanol, an environment-friendly fuel. Being an oxygenated, it contains high percentage of oxygen, which helps combust fuel more completely and reduces vehicular injurious emission. The government blend has made blend of fuel petrol with 5% ethanol mandatory with effect from 1st October 2003 in the 9 states and 4 union territories. So here what they are trying to explain to you is that ethanol is a very safe option when it comes to the fuel, right? It does not produce any residue or smoke when it is burned, and it is a clear fuel. So that is why it is being used in the combination with gasoline and petrol worldwide. Introduction to the production process of ethyl alcohol. Ethyl alcohol has been produced on a large scale for centuries. However, much study could not be accomplished because of hazards on human consumption. In 1865, Alcohol Act was passed, thereby free sale of alcohol after its denaturing by adding methylated spirit was allowed. So, most of the time, people misuse or abuse alcohol. They use it for drinking purposes, okay, and as a liquor. But it was not supposed to be used in such a manner. Then the denaturing of alcohol was done. Early production was primarily used for human consumption, and denaturing was done with the addition of methylated spirit. But today, apart from being used for human consumption, it is also used as universal solvent and as chemical raw material for the production of other industrial products. Along with gasoline, it is used as motor fuel. Because of the above utilities, the demand for alcohol increased enormously today, which led to establishment of many distilleries throughout the world. Ethyl alcohol is produced beside yeast by a large number of bacteria and fungi also. So here is a list microorganisms generally employed in alcohol production, like Clostridium bacteria, okay, thermohydrosulficulum, extremely thermophile, and Clostridium thermophilum, thermophilic, hydrolyzes cellulose, and so on. The list goes on and on. And yeast, for example, Candida species, uh, Pseudotropicalis or C. tropicalis, ferment xylose, Candida species, ferment xylose and cell bios, and list goes on and on. And filamentous fungi, Fusarium species, ferment xylose, Monila species, hydrolyze cellulose and xylem, Mcqs species, ferment xylose and arabinose. So, apart from that, in early days, the yeast was only used. Saccharomyces cerevisiae were used uh, for the fermentation purposes. But as the time passed and the research progressed into further scenario, the scientists have discovered some more fungi, bacteria, and yeast who also carry out the fermentation process in a very comfortable manner. Similarly, besides uses of molasses, lignocellulose can also be used as a substantial substratum. So here you can see the table that uses the global production of lignocellulose waste. Africa, cereal crop 165, pulse crop 9, oilseed crop 11, and plantation crop 34. And when it comes to India, India produces 240 cereal crops, pulse crop 16, oilseed crops are 14, and plantation crops are 88. So all around the world, the production is very high, and all these things can be used for the alcohol production purposes. So these things can readily be used, and they can be utilized for the alcohol production. However, apart from fermentative production, it is also produced by chemical processes, primarily by catalytic hydration of ethylene. So, chemical conversion of lignocellulose by different fermentation are shown in the table. Like fermentation procedures are different. Batch process, continuous with cell recycling, continuous with cell immobilized yeast cell. So here you can see that batch with sugar concentration, cell concentration, ethanol concentration, specific productivity, volume specific productivity. So it goes according to the scenario. So scientists are discovering out new methods and new ways of producing ethyl alcohol. The whole process of bioconversion of lignocellulose into valuable sugars and other substances have been illustrated in the figure. Like agriculture waste and forestry waste are just known to be converted into lignocellulosic biomass, and it is separated out as lignin and reducing equivalent. And then further steps, it is they are subjected to the fermentation procedures, and extracellular enzymes and cell biomass are separated from it. Then fermentation, solvent and organic acids are produced because of that. And moving ahead, the cell biomass is further converted into cellular enzymes and uh, feeds. So, integrated approach for bioconversion of lignocellulosic waste into valuable products that have very good market value. So, fermentative production of ethyl alcohol. On commercial scale, ethyl alcohol production process consists of four steps. So it has basically the four steps, and we must see these four steps. What are those? They are inoculum production, that is essential for the procedure of alcohol production. Okay, what species that you are going to use for the procedure and that will produce alcohol, what type of species, Saccharomyces cerevisiae you are going to use or some other bacterial species, right? Fermentation process. Production medium. So, what type of sugar-based medium that you are going to use? That very particular uh, fungi, bacteria, or yeast that is present in your inoculum, that is going to utilize the uh, medium that is sugar-based. So after that fermentation process, how you are going to carry out? Mostly it is an anaerobic procedure, and it has to be carried out in the absence of oxygen. So it is extremely necessary that everything should go on according to the protocol. And fourth step is harvest and recovery. When the fermentation procedure is complete and everything is up to the mark, then the things are subjected to the distillation unit. So first, production of inoculum. Both yeast and bacteria are used for the production of ethyl alcohol. Right. Among the bacteria, the most widely used organism is Zymomonas mobilis, and among the yeast, uh, Saccharomyces cerevisiae and Saccharomyces carsbergenesis. Certain species of Candida and Mcqs are also used, depending upon the raw material available for ethanol production. So, the species of bacteria, yeast, or fungi is selected depending upon um, the media that you are going to use. Okay, and what we are trying to do here is that we are going to select some sort of species that utilize a lot of media and does not leave anything behind, and it substantially inculcate a lot in the production of alcohol. But high yield uh, and alcohol tolerant strains of Saccharomyces cerevisiae are usually employed. Okay, so alcohol tolerant strain means when they are going to use the process and alcohol is being produced, most of the bacteria and yeast or fungi species are not tolerant to alcohol and they die. So we also have to look into the context that species that we are going to use also be very much tolerant towards such uh, treatment when it comes to alcohol production. Details of process for production of ethyl alcohol from different substances are depicted in the figure. High yield strains of yeast are generally used for commercial. production of ethanol. These are developed by genetic selections. The strain of yeast that may be used for industrial production should possess the following characters. So, what should be the character that yeast that has been selected for the process of fermentation? What are the characteristics it should be having? It should possess uniform and stable biochemical properties. So it should not behave in some sort of funny way or unknown way, and it should be stable and should have very well defined biochemical properties. The ability to ferment a broad range of carbohydrate substrates rapidly. Okay, so it it should not be not only be stable, but also be able to ferment a broad range of carbohydrate substrates rapidly. Uh, it should yield large quantities of ethanol. Okay, it should prepare a lot of ethanol when it comes to the process of fermentation. It should grow fast and should have high osmotic tolerance. Because when it is growing fast and it is also producing alcohol, then osmotic tolerance is highly recommended because that very media will have a large amount of alcohol and very less amount of liquid or water in it. Low levels of byproducts such as acids and glycerols. So one must all see all these five qualities are readily present in that very strain of species that is being used for industrial production of ethanol. It should be tolerant to higher concentration of alcohol. This is also very important. It should possess high temperature tolerance because a fermentation process is an anaerobic temperature and it requires high temperature so that the yeast can readily convert sugar into alcohol. High cell viability for repeated recycling. It is also one of the most important aspects. Appropriate flocculation and sedimentation characteristics to facilitate cell recycling. So, carrying on to all these nine properties, what else we have to look into the matter? However, the choice of microorganism employed in large-scale ethanol production depends upon the type of raw materials used. For example, Saccharomyces cerevisiae is employed when starch or maltose is used as raw material. When whey or sulfite based liquor is used as raw material, Candida utilis and Candida um, albicans, respectively, is employed in the fermentation process. Production of ethanol from starch is shown in the figure. So, what they are trying to elaborate to you is that the selection of the species is somewhat dependent on the selection of the raw material that is employed in the fermentation process. Okay, so your selection for raw material will readily define the species that you are going to select for the fermentation process. Inoculum production process. How you are going to produce the inoculum? A suitable pure strain of yeast is inoculated into a test tube containing approximately 10 ml of sterile medium. Right? A suitable pure strain of yeast is mixed in a test tube containing 10 ml of sterile medium. It is incubated at 28 degrees centigrade to 30 degrees centigrade till sufficient growth of yeast takes place. So, inoculum is a very small portion of medium that is prepared and in which the yeast species readily mixed, and it is kept at the temperature that is supported, that maximum supports the growth of yeast. And the temperature is maintained from 28 degrees centigrade to 30 degrees centigrade until unless sufficient growth of yeast takes place. The medium employed for the preparation of inoculum and the fermentation process is generally similar. Okay, so this type of medium, uh, what type of medium is employed for the inoculum uh, preparation and fermentation process is generally very similar, right? After sufficient growth of yeast occurs, the culture from the test tube is transferred to a flask containing approximately 200 ml of the medium, right? So, sufficient growth of yeast is very important, and the culture test tube is transferred to a flask, approximately 200 ml of the medium that it should be containing. So, see here they are proceeding step by step. First, they are culturing the yeast species in a animal test tube. Then, after the sufficient amount of growth has have occurred, then they are going to transfer the medium to a 200 ml flask that is also containing medium. The flask is incubated at temperature 28 degrees centigrade to 30 degrees centigrade until predetermined cell mass is formed. Fully grown culture from the flask is transferred to a glass container containing 4 liters of sterile medium and is incubated at 28 degree to 30 degree centigrade temperature till sufficient growth takes place. The culture from the glass container is finally transferred to a small seed tank containing 40 gallons of sterile medium. Then, seed tank is to be near the fermentation tank. The culture broth after incubation will be ready for inoculating into the production tank. A large amount of yeast culture ranging from 8 to 10% volume of inoculum is required in industrial production of ethanol. So, if you are using 10 liters of medium, then you require 8 to 10% of inoculum that should be prepared, right, according to the volume, total volume of medium that is employed for the ethanol production. To achieve rapid growth of yeast and large amount of cell mass, high degree of aeration and agitation of the medium is required. So sometimes continuous aeration and agitation is required. The pH and temperature are adjusted at 4.8 at 28 to 30 degrees centigrade, respectively, during the growth of the yeast. Preparation of the medium. This is also an important part. The composition of fermentation medium plays an important role in achieving optimum yield of ethanol. It should be prepared in such a way that it contains all the sources of material that promote optimum growth of yeast and optimum yield of ethanol. So you must prepare the medium in order to show and result maximum growth when it comes to alcohol production. Generally, the medium should possess carbon sources, nitrogen sources, and growth factors. There are several other factors that are also important that we are going to discuss. Our carbon source, right? Different varieties of carbohydrates, uh, which are produced as waste products in agriculture industries, can be used as carbon source. They are grouped into the following categories depending upon their chemical nature. And what are their chemical natures? Starchy material. Okay, the first is starchy material. So, what are the things that are included under that very term? Potato starch, cereals like oats, wheat flour, and corn starch. These are the starchy material that are used for the fermentation purposes. Saccharide material, and what are the things that are included under this term? Fruit juice, whey, molasses, and hydro oil. Okay, cellulose material, sulfide based liquor, lignocellulose. Okay, all these three different types of materials are significantly used for the alcohol production. Molasses is generally used as the main carbon source in the preparation of fermentation media. However, the cane molasses, sugar cane molasses, is used in India because of its availability in large quantities from sugar industries. Because in India, sugar cane is cultivated on a very large scale, so that is why it is readily used in large quantities for the production of ethanol. The raw material indicated above require pre-treatment in the form of saccharification. They are put to hydrolysis during saccharification, due to which easily fermentable sugars like maltose and glucose are formed. The optimum sugar concentration should be maintained between 10 to 18% in the fermentation medium. If beet molasses are used, biotin should be added to meet the biotin deficiency. If cane molasses is used as a carbon source, its sucrose concentration should be reduced to 10% by addition of distilled water, which is called a wort. Higher sucrose concentration affect the growth of yeast adversely, while lower sucrose concentration makes the fermentation process uneconomical. Now, in recent times, lignocellulose as sucrose of carbon proved to be more economical and sustainable. Most of the plants after death are subjected to the process of decomposition, releasing fermentable sugars. The large amount of lignocellulose out of waste agricultural products can be converted into sugar by enzymatic hydrolysis. Annual production of these lignocellulose is around the world is given in the table. So, integrated process of conversion of lignocellulose waste into value-added saccharides is presented in the figure, and ethanol production from different substrates is depicted in the figure. So, what we see, the whey and other dairy products, and they are subjected to deprotonation, and eventually they reaches the fermentation batch, semi-continuous, continuous for the ethanol removal. And starch crops that are cereal, roots, or tubers in nature, then they are gelatinized. Saccharification procedures takes place to convert to glucose and maltose with the help of enzymes, maltodextrins, and then they are subjected to the fermentation procedure. Sugar crops like beet and sugarcane, and their juices are extracted out of, that is, pure form of sucrose. Then it is also subjected to the fermentation. Now, lignocellulosic material like wood chips, sawdust, straw, waste paper, etc., and pre-treatments that readily used for mechanical size reduction and delignification that produces lignin. Then cellulose and hemicellulose are seen that leads to acidic or enzymatic hydrolysis to glucose, cell biose, xylose, and arabinose, etc. So fermentation medium supplemented with nitrogen, phosphate, vitamins, etc. Everything is taken care of. Then fermentation byproducts, animal feed, etc. are also used. Then it is subjected to distillation after the whole fermentation procedure is complete. 95.6 volume by volume ethanol and azeotropic distillation 99.8% volume by volume ethanol is produced. So this is the main industrial ethanol production from various substances. So it is not only the sugar cane based production of ethanol, but readily many different types of materials that are now currently being used in order to produce the ethanol. So agriculture waste, forestry waste, then they are subject to uh, lignocellulosic biomass, lignin, reducing equivalents, right? Then lignin is used as extracellular enzyme fermentation procedures. They do produce extracellular enzymes and cell biomass. Then fermentation subjects to solvent and organic acid production. Then cell biomass is used as cellular enzymes and feed. So this is an integrated approach for the bioconversion of lignocellulosic waste into valuable products. Nitrogen source. Variety of inorganic and organic nitrogen compounds may be used as nitrogen source in the medium preparation. So nitrogen sources are also very important. Ammonium sulfate is generally used as nitrogen source. Generally, 0.15 gram of ammonium sulfate is added to 2.5 gallons of molasses. The concentration of nitrogen should be carefully maintained as indicated above. Excess nitrogen promotes rapid growth of yeast cells and decreased production of ethanol. Growth factors. As most variety of molasses, for example cane molasses, contain suitable concentration of growth promoting substances, hence there is no need for addition of any growth promoting substances separately in the preparation of the medium. pH. The pH of the medium is adjusted to 4.8 to 5.8 by adding sulfuric acid or lactic acid. As the medium becomes highly anaerobic at this range, the pH this inhibits the growth of contaminating bacteria naturally. There is no need of sterilizing the medium. Further, anaerobic fermentation of alcohol discourages growth of many of microorganisms, and most of them are not at all tolerant to anaerobic conditions and exposure to uh, alcohol. However, pasteurization of medium can be done if you want to achieve somewhat more secure methods for the production of alcohol. Fermentation. Ethyl alcohol can be produced by any one of the following processes: batch fermentation, continuous fermentation with cell, and continuous fermentation with cell recycling. So, It depends. The most probably and most widely used method for alcohol production is batch fermentation because readily all the things are mixed together. Medium is prepared very nicely, and inoculum is also well maintained. Then it is added, and what is seen that when the medium it starts to make small bubbles, it signifies that the process of fermentation is going on. And when the bubble making procedure ceases out, then you can say that uh, the fermentation procedures are not going on, or the fermentation process has been completed. Now the microorganism that has been employed is not at all utilizing the sugars to make ethanol or alcohol. So production of inoculum, both yeast and bacteria are used for the production of ethyl alcohol. Among the bacteria, the most widely used organism is Zymomonas mobilis, and among the yeast, Saccharomyces cerevisiae and Saccharomyces bergaensis. Certain species of Candida mucor are also used depending upon the raw material available for the ethanol production. But high yield of alcohol tolerant strains of Saccharomyces cerevisiae are usually employed. So, high yield strains of yeast are generally used for commercial production of ethanol. These are developed on the basis of genetic selection. The strains of yeast that may be used for industrial production should possess the following characters that we all know. So, composition of fermentation medium plays an important role in achieving uh, optimum yield of growth. Right, it should be prepared in such a way that it contains all the source of materials, right, and it should also carry the uh, carbon sources in a very pre-decided manner and in a very controlled way, right, and optimum sugar concentration should be maintained 10 to 18% in the fermentation medium. If beet molasses are used in biotin deficiency medium, then it should be reduced to 10% by the addition of water. So when it comes to nitrogen sources, a variety of inorganic or organic nitrogenous compounds may be used as nitrogen source in the medium preparation. Ammonium sulfate is generally used as nitrogen source. Generally, 0.15 gram of ammonium sulfate is added to 2.5 gallons of molasses. The concentration of nitrogen should be carefully maintained as indicated. Excess nitrogen promotes the rapid growth of yeast cells, decreasing the production of ethanol. So growth factors, as most varieties of molasses, for example cane molasses, contain suitable concentration of growth promoting substances. Hence, there is no need for addition of any growth or promoting factors in the preparation of medium. And pH is adjusted to 4.5 and in between 5.8 readily by adding sulfuric acid and lactic acids because these things they readily balance the production of ethanol. So there is no need, also no need of sterilizing the medium. Further anaerobic fermentation of alcohol it discourages the growth of many microorganisms. However, pasteurization of medium can be done. Rectifier. Just like the analyzer, a rectifier also is also a vertical tower, right? It consists of specifically designed uh, fractionating columns with a number of chambers. The less volatile constituents, the slop and the fuel oil, gradually condenses and are drawn off from a higher point of the still. The temperature at the higher point is still roughly equal to the boiling point. The head products, which are very less, contain aldehydes, forming esters, etc. So these oils and esters they are used in a different kind of production for the production of food grade items, right? And they are utilized for the production of essential oils and other edible products that are utilized in a very different manner. So nothing is wasted in the alcohol production procedures. Everything that is being produced in the rectifier is readily recycled and reused. And these products are also very costly in nature because they are being through such a lengthy process and they are being employed in every aspect. So many people or many companies they readily purchase these esters and oils that are being produced in the process of uh, rectification or distillation. Owing to their greater velocity, pass through the top of the column along with a small quantity of uncondensed alcohol. The higher alcohols are removed from the receiver for every 2 or 3 days. So, applications of ethyl alcohol. It is extensively used in the synthesis of variety of organic compounds and is a universal solvent. So organic solvents they do have a lot of applications in every laboratory for the not only for the process of sterilization and cleaning but also in the chemical procedures and experimentation. Second, as energy source in motor fuels, being a clean fuel, it does not produce any sort of smoke or residues, so it is readily used as a clean fuel. Different byproducts are useful in a variety of ways. So, in the meanwhile, they have already elaborated that alcohol production not only produces alcohol, but it also produces many types of aldehydes, right? Esters. Aldehydes are readily employed for the production of pharmaceuticals. Okay, and many types of aldehydes are extremely beneficial in the production of medicines. Similar way, esters are used for production of many food grade items, right? And oils are used for the production of cosmetics also and production of pharmaceuticals things also. Now coming on to the MCQs, uh, Monascus uh, purpuris is utilized in the production of A. Citric acid, B. Ethanol, C. Statins, or D. Streptokinase. The correct answer is C. Statins. The type of fermentation observed in yeast is A. Alkylic fermentation, B. Lactic acid fermentation, C. Pyruvic fermentation, or D. Alcoholic fermentation. The correct answer is D. Alcoholic fermentation. In lactic acid fermentation, the final electron acceptor is A. Lactic acid, B. Pyruvate, C. Oxygen, or D. NAD. The correct answer is B. Pyruvate. Which of these is not a product of fermentation? A. Lactate, B. Oxygen, C. Carbon dioxide, D. Ethanol. The correct answer is B. Oxygen. So oxygen is not at all included in the uh, fermentation process because it is all anaerobic in nature. If oxygen will enter the whole process, it will convert the alcohol into vinegar, and the whole process will be destroyed. Which of the following is not a vegetable or food-based fermented product? A. Wine. B. Sauerkraut. C. Beer. D. Vinegar. The correct answer is D. Vinegar. One of the most commonly used fermented cereal amongst these is A. Wheat. B. Bread. C. Rice. D. Yogurt. The correct answer is B. Bread. 7. Glucose molecule during the process of glycolysis is broken down into 4 pyruvic acid, 3 pyruvic acid, 2 pyruvic acid, or D 1 pyruvic acid. The correct answer is C 2 pyruvic acid. Fermentation occurs in A. Presence of oxygen, B. Absence of oxygen, C. Presence of nitrogen, or D. Presence of carbon. So the correct answer is B. Absence of oxygen. It is an anaerobic process. The least yield of ATP is observed in anaerobic respiration, B. Anaerobic respiration, A. Aerobic respiration, C. Fermentation, or D. Same in A and B and C. The correct answer is C. Fermentation. Anaerobic respiration by yeast produces A. Carbon dioxide, B. Wine and beer, C. Alcohol, D. All the above. The correct answer is D. All the above. So these are the references from where we have collected the material. And thank you very much for listening to this lecture.